Hello, everybody, and welcome to the European Marine Board's third Thursday Science webinar series. My name is Sheila Heymans. I will be your moderator today. I'm the Executive Director of the European Marine Board. So before we start, just a few housekeeping rules. Please make sure that your name is clearly entered, so when you ask a question, we know who you are. Also, use the Q&A, which should be at the bottom of your screen, to put in any of your questions, and it would be helpful if you add in there who you're from, where you're from, which organization, and which country. And then once we've had the talks, I will look at the questions and ask them to the speakers. If you have any technical problems, please use the chat function, and be aware that the webinar is being recorded and it will be made available on our website and on our YouTube channel after the fact. I just want to welcome today the speakers. Our speakers will be our two outgoing Marine Board young ambassadors, Alexandra Hahn and Riwan Leroux. If you guys can put your video on, thank you. And they will be talking about their science in a talk entitled "From Coastal Waters to the Deep Sea, Marine Life Under Environmental Change." So I believe that Alexandra will start and Riwan will follow. We will take the questions at the end of their presentations. So I will stop sharing, Alex, and you can figure out how to stop sharing. There we go. And you can share. Thanks.
Thanks, Sheila, for the introduction. And let me just share my presentation. And I hope you can already see this in the right screen.
Yes, I think so.
Yeah, well then, let's begin, shall we? So I'm very excited to be here, not only as an outgoing ambassador, but also as a PhD student at GEOMA Helmholtz Center in Kiel, Germany. And I'm very excited to talk about my PhD research in local adaptation to salinity stress. And before I start with my actual science, I want to introduce the concept of local adaptation to you so everyone is on board and knows what I'm talking about. So imagine there's a marine species that I show here as a fish, and then this fish has a thermal tolerance. So here I show temperature on the x-axis and then the performance of the fish as measured in like a fictional experiment by this curve. So the top of the curve, this is the optimum temperature for this fish, but it can survive in all the range that the curve covers. So if we now imagine that this fish species occurs close to the equator, and then we can imagine that the curve shifts towards warmer temperatures, and this fish species evolves to tolerate higher temperatures. And then conversely, if the same fish species would occur close to the poles, then this performance curve would shift to lower temperatures. And if we observe this genetic difference, and there are distinct populations of one species where an individual of the species performs best at their habitat where it naturally occurs, so the cold polar species performs best at cold environments, and the warm equator individual performs best at equator close temperatures, then this is called local adaptation. And this concept is very interesting for a variety of reasons. First, from a management perspective, you have to treat a species differently if you have distinct populations with distinct habitats. Now, of course, no one would manage a polar and an equatorial species in the same context, but this local adaptation phenomenon can also occur on much smaller scales. So, for example, the inside of a fjord versus the outside of the fjord could already be locally adapted populations. And then, of course, you cannot treat them as one big pool of animals. But the reason why I'm interested in local adaptation is that we can use this gradient in space, this environmental gradient, to understand the gradient in time. And what I mean by that is that global change is transforming our oceans, so the temperatures are getting warmer. And if we now find that there is local adaptation, then we can use this knowledge to infer adaptive capacity. So for example, this polar species could adapt to tolerate warmer temperatures and then be more closely in their thermal range to the present-day equator species. And this one could even further adapt this individual of the same species to tolerate higher temperatures, which could ensure the survival at this location. And I'm not looking at this for temperature, no, I'm not investigating local adaptation to temperature, but instead I'm looking at salinity. And to do that, I'm researching the Baltic Sea. So the Baltic Sea is a very extreme environment. It's a very small sea with little exchange with the adjacent North Sea, so there's very little water inflow, which makes it a very brackish water to freshwater environment compared to the North Sea, which is fully marine. And this puts a lot of stress to a lot of organisms that want to establish in the Baltic Sea. And that's because as a marine animal, your environment has the right ion concentrations as the inside of your cells. So you're losing a bit of water and ions are coming in through your skin, but you don't have to actively regulate that much. While if you live in a freshwater environment, as a fish, then you are losing a lot of ions and you have to actively transporting back in. And not a lot of animals can do this switch from a marine habitat to a freshwater habitat. So that's very difficult. And this is why the Baltic Sea is a very challenging environment to live in, because organisms, if they manage to live there, they have to either constantly compensate or they have to adapt to this novel condition. And this has been shown for phytoplankton, for mussels, and for fish. And we hope that by understanding adaptive capacity, we can predict and understand how future climate change might affect organisms in the future. And I'm studying this not on fish, but instead on copepods, and I studied on the species Acacia tonsa. Now, if you don't know what a copepod is, this is what you can imagine for the rest of the talk. So copepods are tiny crustaceans, and they're a very fascinating and valuable study organism for several reasons. First, they have a global distribution. So there are roughly 15,000 described species that live in any body of water than you can imagine. And also the species I'm working with, Acacia tonsa, can be found all over the world. So what we find here is also relevant for other locations. Then copepods in general have a very high ecological importance. They feed on phytoplankton, and then they transfer the energy to higher trophic levels, which often is fish species that are also commercially used. So they're the basis of the food web, and we want to know what happens to them, also for the sake of other species. And then from a practical standpoint, the species I'm working with is very small, so we can take them to the lab and we can culture them there. They have a very short life cycle, which helps us to work on experiments that are not possible with fish in the same manner. And the question I'm asking my PhD is Acacia tonsa locally adapted to the salinity in the Baltic Sea? And to answer this question, we have three different approaches. First, I'm conducting fitness experiments at low salinities. Then we're looking at gene expression in response to low salinity stress. And we're looking at population genomics, so we compare the genetic information of copepods from different locations. And in my talk, I will focus on the first two of these. So let's start with the physiology, so the fitness experiment part. And there we ask the question, is there a difference in fitness at low salinity? And to approach this question, we sample copepods from the North and the Baltic Sea, and then we transfer them to the lab. And there we culture them for several generations at the same condition. And that's, if you think back to the local adaptation concept, we want to get rid of environmental variation. And then if we do experiments with these common gardened, so these lab animals, and they still show differences in their performance, then that's because they're genetically different, and that means there's local adaptation. So then we expose them to low salinities, the North and the Baltic Sea copepods, and we just check how many of them can survive this stress. And what do we expect? So we expect the copepods that are adapted to low salinities to also show higher survival at these extreme low salinities, while the North Sea ones show lower survival. If you think back to the curve that I showed at the beginning, and you imagine Acacia tonsa as a marine species, so the North Sea performance curve would be very similar to the normal Akasha-Tonsa fitness curve, and then the Baltic curve would be shifted. So if we then test at these very extremely low salinities that are towards the edge of the distribution of Akasha-Tonsa, then we expect the Baltic ones to perform better. And these are the actual populations that I'm using to the right side. And then I measured mortality after 24 hours. So you see on the x-axis the salinity that I used in practical salinity units. 5 is sort of the distribution boundary. So this is the lowest where they can survive and reproduce and be generally happy. And then we go as low as freshwater, which is at 0. And there I score the animals at either dead or alive. So you see a little point. It means that there was an observation. The darker it is, the more animals survived. And then I can fit a curve through this. That looks somehow like that. And you see also this dashed line. And this is the point where 50 of the animals are alive or dead. We can follow this to the x-axis, and then we get a salinity value that explains that this salinity, half of the animals are able to survive after 24 hours. And this is around 1.4 salinity units, so very low for the Baltic Sea. And for the North Sea, this curve shifts almost 1 practical salinity units, which means that the Baltic copepods can survive lower salinities compared to the North Sea copepods. So the next question that we have is, what are the mechanisms behind the salinity tolerance? And to answer that question, we have to go into transcriptomics. And there ask the question, how does gene expression differ between populations? And also, is there a common or unique response to low salinity stress between the two populations that we're looking at? So to explain the concept behind transcriptomics, you're probably all aware that the DNA is containing all the genetic information, so it's a blueprint, but you only need little parts of that at a given time. So you're not reading all of the DNA at all time. That wouldn't be feasible, but just little parts. And these transcripts are called RNA, and they're often coding for proteins. And the protein in that context is the phenotype. So what we observed before was a phenotype. We measured fitness, and now we're looking at the step that comes before. We're looking what is transcribed from the DNA. And transcriptomics in that context is the analysis of all RNA at a given time. So it's a snapshot that helps us understand what processes are active or inactive at this given moment. And what we are testing is the stress response to low salinity. So we use a control, that is the culture conditions, and a low salinity that's supposed to induce stress to the organism, so we know what they do to counteract that stress. And we measure at different time points to also track this. So we measure before we expose them to these conditions, then we measure shortly after, and then we measure a day later to kind of see if something changes with time. And what do we expect again? We expect the Baltic copepods to be adapted to low salinities, therefore show less signs of stress, while the Baltic North Sea conditions would favor more stress in these animals. And if you think back to the strategies that organisms can have to counteract low salinity stress, we would also expect to see a lot of active ion transport to counterbalance the loss of ions that is passively occurring through the cells. And then what we do first is we look at all the transcripts that are differentially expressed. So we compare controls to the treatment, and we just look how many of them are there. And what you can see is that there is a lot of transcripts at the second time points after 24 hours in the Baltic Sea, a lot more than at any of the other time points. And because this is the difference between control and low treatment, this means that the North Sea copepods seem to struggle. They have to express more genes. There's more happening in response to the stress. But you also see that there's quite some overlap, so genes that they share, which could be a common strategy, so some ion transport, presumably. And to do that, we don't look just at numbers, but also at the functions of the genes that are expressed, and we look if any of these genes are overrepresented to what we would expect in like a normal state, and then we can assign them and group them by their functions. And we can also first see if one of the populations has higher expression. You see down there, this is the control state, so it's very steady over time. But you see that the Baltic ones, all these shared genes, they have them expressed just a little bit higher, which could also mean that they're just more efficient. They're doing what they need to survive a little bit more, and this is why we don't see them dying as fast as the North Sea animals. And then we look at the functions. You don't have to read everything that's on the y-axis. I will sum this up, but I'm just showing this to you to show that there's a time-dependent response. So what happens at this initial stress point after 3 hours is very different to what happens after a day. And also you see a lot of red at this first time point. This means that there are a lot of upregulated genes, so that are more present in comparison to the control, while at the second time point, there are also some that are downregulated, so they're less present. So it seems to be a little bit more nuanced. And after 3 hours, we have a lot of metabolic and neurological behavior regulation. So there seems to be some initial response to try to avoid the stressful situation and to remodel and prepare. But there's also ion transport, which is something that we expected, and pH regulation to counteract this osmotic salinity stress. And then after 24 hours, we have a lot more other transport functions, also of larger molecules, such as lipids. And we have a lot of processes that aim to establish homeostasis to so get to an equilibrium state where it's not as much active ion transport is necessary anymore. And then we also look at processes that are unique to the Baltic or the North Sea. And we see that the Baltic Sea has very few additional functions and none at the first time point. And because, again, this is a deviation from like a normal steady state, this means that there's less salinity stress in this population. And again, it's a sign of local adaptation. While the North Sea population has a lot of additional functions, and a lot of them are in blue, which means that the genes that drive these functions are mostly downregulated, which means they're suppressed, they're less present than in the control. And this, if you look at what they do, there's a lot of metabolic and developmental functions that are being suppressed, likely to conserve energy. So this is a sign of a struggle to comply with the low salinities. So they try to save energy that they can then invest in all these ion transports that we've seen before. So to sum up what I've just explained to you, we saw differences in gene expression and in fitness, which are a sign of local adaptation. But we also saw that both of these populations have a very high tolerance to low salinity. They both have a shared osmoregulatory strategy. And if you think back to the first experiment that I showed you with the fitness, they both can, at least on a short-term basis, survive quite low extreme conditions. And then what I'm doing next is I'm using a population genomic study to identify the genetic basis of salinity tolerance and also verify the findings we've seen before. And for this, we will not only include one station in the Baltic and one in the North Sea, but we use the whole environmental gradient to better get a grasp of what is happening. And what does this mean for copepods in a changing ocean? So first, the high resilience to environmental stresses that we saw in both of these populations could ensure at least a short-term survival and then buy some more time for adaptations to occur, because of course this is also a process that needs some time. And that the adaptive potential that we saw in the Baltic population could help copepods to cope with future environmental change. And I only did this in salinity, but there are other studies that also show that copepods can adapt to temperature and to ocean acidification. So basically to all the stresses that will occur in the future with ocean change. And with that, I want to thank you and all my collaborators, and I will give the floor to Riwan, who's talking next about deep sea.
Thank you very much, Alex. Very interesting. So Riwan, I think you also will share your screen, right?
Yeah.
And a reminder for
hello,
everybody.
everyone.
Sorry, Riwan, just quickly while you put your screen up, a reminder that if you have any questions for Alex, please put them in the Q&A and we will take them afterwards. Thanks.
So hello, everybody. I'll go deeper now in the deep sea, but still looking for the anthropogenic influence on the ecosystems. So I will present my work about how scientists themselves might exert pressures studying the deep sea, a work I've done in my postdoc position at Ifremer with my colleagues. So first, what are the anthropogenic pressures in the deep sea? So I distinguished two kind of pressures. First of all, the global pressures with the climate change, as Alex said, and the temperature rise, acidification, or the deoxygenation of the ocean. And we have also the pollution with contaminants and plastics, which comes from the coast and spread into the ocean. And then we have the more local pressures with the energy industry and gas and oil extraction, for example, that occurs in the deep sea at several thousands meter depths. And we have also the pressure of the fishing activities. In the future, we will also have two different kind of pressures from the human activities with the engineering climate intervention. So for example, the marine carbon dioxide removal, which consists in growing some organic matter at the surface, stocking carbon, and then putting it down into the deep sea to stock the carbon and reduce the concentration of CO2 in the atmosphere. And we will also maybe have the mineral exploitation of several ecosystems in the deep sea. So the nodule resources in abyssal plains, the cement ecosystems, or hydrothermal vents. These three ecosystems contain a lot of different attractive metals like lithium, gold, rare earth elements. And you can see on the map the different source of minerals in the deep sea that are of interest for the industry. In my talk, I will talk about hydrothermal vents, which is also a source of minerals, like I said, but as you will see, I will talk about a different kind of pressure. So the hydrothermal vents are ecosystems that were recently discovered, or quite recently, since we discovered them in 1977. And they are really particular in the deep sea because they are really characterized by high biomass ecosystems, like you can see here on the video. These Atlantic hydrothermal vents are covered by mussel beds, and you can see also large white filamentous material microbial mats. And these ecosystems are fueled by a lot of chemicals that enable the chemosynthesis in these ecosystems. So these ecosystems are able to produce organic matters from the environment through the fluids that are expelled here, which are really rich in minerals due to their travel in the oceanic crust. And these ecosystems form the seafloor massive sulfide deposits that are of interest for the industrial exploration and exploitation. On the video here, you can see the shimmering on the frame. That's the fluid that are expelled in the chimney, and that fuel the chemosynthesis. So the question is, will be some future mining exploitation at inactive vents? Maybe, maybe not. It's quite a huge question since it's involved the international relation and legislation. But what about active vents? In these ecosystems, so I told you there is a lot of life and a lot of knowledge to discover. And currently, there is one direct pressure at hydrothermal vents. These are the research activities. So my postdoc and my project was about determining if these research activities could pressurize the ecosystem enough to create impacts on these ecosystems and modify the equilibrium, the community composition, etc. To do that, I focused on the Lucky Strike hydrothermal vent field, which was discovered in 1982, and it is located in the marine protected area of the Azores. So it's kind of a unique opportunity to study this question because this vent field was studied almost annually since its discovery and hosts a deep sea observatory since 2010. And since its discovery, we went there with 39 oceanographic cruises, representing more than 300 dives and thousands of scientific investigations. So the idea is to census all these activities and determine if we can detect an impact from that. So the research activities we were focusing on was mostly these ones. So we have the samplings on the left with the suction sampler and the more classical sampling that you can see on the bottom left. We have also the measurements, the moorings, the fluid samplings, the sediment samplings. The 4 pictures on the right depicts the different instrumentation we deploy on the bottom of the vent field, and each deployment required to have some metallic weight to deploy them on the bottom, and each time we need to recover them, we just lose the weight on the bottom. So these activities also are a source of trash on the bottom. And finally, there is also the submersible presence by itself with the light in an environment without any light, and noise and vibration also. So to recover all these activities, the location, the date, etc., it was a really hard work to go 30 years ago until now. But we succeed in recovering the most of these activities with more than 3,000 samples, more than 3,000 hours of submersibles trajectories, and a lot of metallic deposition on the bottom. So we did this census, but we still have to do some calibration to take into account other kind of activities, but it's kind of difficult because some activities are accidental, like the one you will see on the video where the submersible will just go back and with the propellers and put some water on the chimney that is quite recent and fragile. So you will see that the chimney will collapse, which is not, we can't take all these fortunately rare events in our analysis, since they are not quite reported always. So once we have all these activities location, we weighted them to be able to say, okay, then the collapse. So it's a bad thing, but like I said, it was a recent chimney. In between 1 and 3 years, the chimney will grow back at the same size. So we weighted all the activities, asking for experts to give their opinion about which one is more impactful than the other. And then we construct an index to integrate all these activities and to pull them to be able to map this index, this unique index, to characterize the research pressure into the environment. So here is the map. On the top, you can see the whole Lucky Strike map with the pressure index, and you can see some zoom on the bottom. So I will just say that most activities are occurring at the active vents and much less in the peripheral areas. And among the active vents, there is a lot of differences between the pressure level between these active sites. On the right, you can see the Tour Eiffel edifice, which is the most studied edifice. We go there every year. We study the ecology, the chemistry, the physics, etc. So this is a lot of research pressure. And you can see also to the left a little dot here. Here, that is one of the instrumentation platform of the EMB Abassadors Observatory. So it is peripheral, but there is a lot of research pressure too here. So we did analyze these pressures for the different active sites. And to show you the contrast between the different sites, I will show you this figure. So in the y-axis, we have the pressure index. And in the x-axis, we have the years. So you can see the temporal evolution of the research pressure among the different sites at this vent field. So in green, the sites that were not really pressurized by research activities. There is quite a lot. And then we have the blue ones, which were relatively pristine before 2010, and then the research activity grew. And I will discuss about it why. Then we have the yellow one, the 3 sites that were more pressurized, but remain similarly than the blue one in terms of overall research pressure. And then we have 2 other sites, the Tour Eiffel edifice I mentioned before, and Montségur edifice, which encountered a lot of research pressure and almost from the beginning of the exploration of the vent field. So we can detect an effect of the observatory installation in 2010, especially for the blue and yellow sites. It means that when we installed the platforms and the observatory in 2010, we started a multidisciplinary and time series survey. And that's why a lot of active sites were experiencing an increasing research activities, but this was most likely monitoring activities, a few fluid sampling, etc. On the contrary, Tour Eiffel or Montségur edifice were the focus of experimental studies, definition experiments, and impact experiments. So they were a lot more pressurized. So here is the current results I have, but as you can see, I did not talk about impacts, but just about pressures. So the next steps will be to go from the pressure to the impact to actually see if we have some, for example, decrease in terms of biodiversity, etc. So to do that, we will use 3D reconstruction of active edifice that I filmed this summer. And so we have a lot of edifices to annotate and to see if there is differences in terms of, for example, surface of mussel beds, etc. And we will use these data to understand if the research pressure has significant influence on these distributions in terms of mussel beds, but other assemblage and biological distributions too. And we will need to disentangle also the natural viability among these active vent sites, because we know that if the fluid is stronger or if there is more exits on one edifice, there is also more biomass, for example. So it will be kind of difficult to detect the impact of research activities due to this natural variability, but we will try it. And the idea is to use these results to improve or to give advice to the Azores government, since we are in collaboration with them to give them the maps and the results to improve the good practice in the research in the deep sea, and especially at Lekesrag Venfield. So, like I said, we will try to do that, and fortunately, we have a lot of 3D reconstruction to analyze. So almost 17, I think. So we are confident that we will be able to disentangle natural variability with the what we expect to be a small signature of the research activities impact, but still stay tuned for the further results. Thank you.
Thank you, Riwan. That was very interesting. Maybe you can stop sharing your screen.
Yeah.
And then if Alex, if you want to come back, then maybe we can take some questions. I see that, hold on, that I see that we have one question in the Q&A. This is from Sahar Stevenson-Jones. Specifically, I guess it's for Riwan. It says, what a fantastic research question. Maybe we should be asking this question in all locations that we conduct research. It's not really a question.
Yeah, I think this is the way we are going to. A lot of institutes and funding organisms will ask that in the future and are going to ask that now. So yeah.
Yeah. So, but I have a different question. Do you think that we shouldn't be doing this research at all? Because I mean, there's an impact, but it's really impossible. Well, understanding how it is, what the impact is compared to the natural variability is one thing, but also understanding what the impact of this is compared to another activity is really what we need. We need to know the impact of research versus the impact of fishing, or versus the impact of mining. Otherwise, it's just looking at something in isolation and you know, something to hit just over the head with all the time.
Yeah. For the part of should we have research activities in the deep sea, my answer is not cannot be yes or no, but of course I love my job, so please do research in the deep sea. But I think it's an ethical question we need to think about, and I think the good way to think about that would be to follow some good practice, to be assured that the sample we take are used and valorized and shared and available, etc. And about the comparison between research activities and other kind of pressures, yeah, definitely the idea. I believe the impacts we will maybe detect will be really low, because we see the edifices each year, and we know that the variation is not really visible in a way that obvious, but we can see some clues of this impact. But to compare that to other pressures, we would need to determine which kind of pressure occurs directly on hydrothermal vents at active vents. And for now, there is no mining activities at active vents, and I believe there will be no mining activities at all at active vents, because of course it's a precious ecosystem. For the fishing, if there is fishing at other hydrothermal vents, it's accidental. So there is no really direct anthropogenic activities in the vents apart from the research. So yeah, it would be interesting, but hard to do.
But you also have to be, because you're focusing very much on the research activities, but we have to always remember these are research activities on active vents versus other uses. And if there aren't any other uses, that's fine. But the way it's communicated, and this is actually really important for any kind of communication of the science we do, is that quite often all they will say is research has a huge impact. That is what they will report. So how do we make sure that that they understand that this is research on an active hydrothermal vent versus a non-active hydrothermal vent versus in a different area? So we really need to be very clear that when this type of work is reported, that it doesn't, it's reported in the right way, if you know what I mean.
Yeah.
Because it's very, the press will really easily want to make this into an easy story, but it's not an easy story. There's many caveats that we need to make sure is out there.
Yeah, I think the communication is firstly for the scientists, but yeah, we need to think of how it can be devoid, and because obviously industrials will be, look, scientists already fucked up all the ecosystems, so we can go there.
Precisely, yeah. And then one last question before I get to questions for Alex. I'm still not seeing any questions. And oh, there we go. We have a question. And this one is also for you, Riwan. So maybe we'll start with that one. Lucia Frachik. Sorry, I cannot pronounce your last name. Visual artist in 2D, living in Croatia. Her question is, I was wondering if I could create new artworks inspired by some of your visuals. Yeah, that's something for you guys to talk about.
Yeah, send me an email. It would be cool.
I think we can take the details here and we can put you in contact as well. Yeah. But now I've forgotten my second question to you. I can't be doing this. But maybe we can come back to Alex. Alex, also with your project, I was kind of trying to figure out how do you communicate the results of what you have. So if I understand correctly, the copepods in the Baltic is more adapted to low salinity and is able to deal with it better. What is the application of that, I guess, from a management perspective?
Yeah, that's a good question. And I also believe someone asked something along those lines in the chat, and I can come to that.
Okay.
Exactly. So I don't know if I communicated that clearly, but they are the same species, right? I'm looking at one species, and I showed that historically in the past they were able to adapt to this changing environment, or to this like extreme environment, the Baltic Sea. And the Baltic Sea is also predicted to further change. So for salinity, there are some models that predict there will be increased rainfalls and more river runoff, which will further decline the salinity there, which could push a lot of organisms to their boundary and like push them further out of the Baltic Sea. So we wanted to know if that's the case for my study species, Acacia tonsa, and now we're like cautiously optimistic that they probably are able to tolerate and also to further adapt to this condition. And then also for the species also outside the Baltic Sea, just showing that they can adapt to very extreme environments is something that gives hope for future change, not only in salinity, but also in temperature, and in ocean acidification, because not all organisms can actually adapt, but they show a very high potential and because they're so important for food webs and like as a food source for fish. And this is very good sign. At least on this end, we don't have to worry, and we can focus more on managing fish stocks and not like destroying them, because their food source for now is probably going to be fine.
I'm going to stop you there because I have a question specifically on that. And it is a little bit about the food web implications, because I'm not sure if being well adapted to salinity necessarily so does that mean if they are adaptable to a specific salinity range, they would still produce the same lipids? Because for food webs, it's one copepod is not another copepod. If they're not fatty enough, then they're not necessarily be as good for the fish. So do you get that kind of information from the genetics, or?
Yeah, so far we don't have that information. Like we don't know if they're the same like nutritional value, and there's probably some differences. But at least we know that they're not completely going away. So there at least is some food to like eat. If it has the same value for the predators is like unclear. No, I cannot tell you.
Because that's really in food webs, that's often the case. And that I think they've even shown those kind of things in the North Sea and in the West Coast, the western part of the Atlantic, the eastern part of the Atlantic, that, one zooplankton species is replaced by another zooplankton species, and they're kind of edible, but they don't have the same food, nutrient value. So then even though there is food there, it might not necessarily help them anyway. So I think it's something, it's good to know that they can survive, but maybe from a food web perspective, we need to look in a little bit more detail. Okay, and then I see the Kushich Shure, I guess is that the question you saw in the chat?
Yeah.
You did not put the question in the Q&A as I asked. Master's student from James Cook in Australia. Why is it important to understand the metabolic regulation of copepods with reference to the habitat they are found in, and how the presence of various stressors in their environment can be replicated within a lab setup?
Okay, so I believe that's two distinct questions. So the first one is like, why are we interested in understanding metabolic regulation? That's just because we're testing the response to stress, and in that case is salinity. So we want to know how the organism responds, and transcriptomics gives us a very like easy window. Like it's easy, like relatively easy to look at all the transcripts instead of like looking at all the genetics changes, or also looking at proteins. Like it's very difficult to look at like protein reactions. So transcriptomics is an easy way also to just understand in general how they do it. Like they have a very high tolerance against various stresses. So we're just like interested in knowing how copepods generally survive. And then the second question about how we can replicate that in the lab. So for my research, I only look at one stressor in isolation, which is salinity. And of course, you can imagine that a research setup gets infinitely more complex if you add more stressors. And that's the interesting part, right? So in the future, it would be very interesting to look at a joint stressor that's like temperature and salinity. So we decrease salinity, we increase temperature. What does that do to an organism? But this would be very more complex, and I'm hoping I can do something like that in the future. But for now, we just focus on one in isolation to just like get the baseline right and just know what happens if you look at one in isolation. Yeah.
Very good. And then another question from you in the Q&A from Frederick Goya. When salinity changes, several other factors also change, like mixing of the water. Where are all the changes in the water related to changes in salinity considered in your study?
Yeah, so we did not consider the mixing, so the stratification. I believe that's what you're referring to in our study. That's of course also something that's going to change, but copepods have to feed in the upper layers of the water, So what will happen in the future, like we were looking at surface salinities because at some point of the day they have to migrate up to these layers. So you have increased stratification, so a layering of salinities in the Baltic Sea. That means you have the low salinity on top, and then you have the higher salinities on the bottom because they're more dense. So that would just mean that the copepods have less like habitat to live in if they cannot cross this boundary layer, but they still have to be exposed to these low salinities, which might be something they will try to avoid, but they cannot because they have to feed on phytoplankton and that grows in the light, so that will be in these upper layers. So this is why we look at the response to low salinities, even though that we're aware that there are also like different layers that are present in the Baltic Sea.
And then I think Paula in our in the chat also asked the question, can we use what you have found in on the ways in which copepods are able to adapt to changes in environmental conditions to help understand how other species could or couldn't adapt? Like, is it species transferable?
It's difficult. Like, it is possible to get some insights for other copepod species, so there are like almost 15,000 of them described, and you can transfer some of that knowledge, but of course it gets increasingly more difficult to apply this knowledge to other species that are non-copepod species. So for now we're just focusing on that. But of course there's like these mechanisms that's still something that's universal, and just understanding this in one system, like you could at least apply the concept somewhere else, even though if you don't know to what extent it will be like correct in another organism.
Yeah. And then Angel, one of our science officers, also has a question for Riwan. Why are the two sites that you mentioned the most studied? Are they the easiest to access, the most active, the oldest? Why are they, Riwan?
So yeah, definitely about the activity. There is some sites that do not have, for example, black smokers or high fluid exits. So I think this is one of the reasons Tour Eiffel was chosen, because it's really an active one with a high temperature, almost 350 degrees for the fluid that exits from the chimney. But this is not the only reason. I believe that it's kind of just convenient because it's a large site, the largest in the Venn field. So it's easier to deploy moorings and do experiments on the bottom, because when we sample, when we do experiments on the bottom, the submersible has to be still, and it's better if it's on the ground. For the Montségur, it's really like 30 meters away from Tour Eiffel, so it's convenient to go from one to the other, because if you want to go with an ROV 300 to travel 300 meters, it will take you like 15-20 minutes. So it's easier to do that. And also this was the first or at least the second sector where we discovered the active sites. So we have data for that since 1984, like 2 years after the discovery.
So I have another question for you, and it is about, did I understand correctly that the EMSO mooring went in like 2010 or something? Or yeah. And so did they put the mooring there specifically because of the site, or was it there for a reason? Okay.
Yeah, they had to deploy, at least they decided to deploy a deep sea observatory for hydrothermal vents, and they decided during the project proposal that it would be Lucky Strike. So they chose this vent field because I don't know why did they choose that, but I believe that the biological community there were really with high biomass. So
Yeah.
it was interesting to have such
and then because you mentioned that there's a more activity around that site, because I guess it gets they go back every year, and do they also do more things there? Like, what if I had to say, is it worth having a deep sea observatory there, or should we not have put it there? Do you think there's a benefit to having it there?
Okay, a little disclaimer: I'm not in the strategy of this project, and I will be on YouTube, so please don't take my word for the truth. But you're right; it was a question about the relevant aspect of having an observatory still after 14, 15 years, and in fact, we answered a lot of questions about the hydrothermal temporal variability in terms of chemistry, physics, biology, etc. So recently, it was really a question about do we keep this observatory, or do we remove it, because it takes a lot of ship time, it takes a lot of team effort, and it's really expensive. So the decision this year or last year was to continue, but the leading team of the cruises had to work again their application to highlight new research questions and new angles. So yes, I believe it's really interesting to have that, because you cannot answer questions without having this temporal series, and above everything, the multidisciplinary aspect of the work, because this is really multidisciplinary cruise where you have all hard science disciplines, but also social science, artists, and it feeds a lot to the community. So yeah, I think it's important, but we have to renew the questions.
Yeah, and I completely agree that in most cases, understanding how things change over a longer time really makes a difference. So it doesn't make sense to move to a different space where again. It's better to continue working in one place. And actually, maybe this is not a question, but a comment that, it's obvious that the stuff that you do cannot be done in the same setting as what Alex is working in. It's not like you can take these animals and stick them in a test tube in the lab, right?
No.
This is not the same kind of science. And so there's different ways of getting at answers. And I think this is a really good way to bring the two things together. So.
Yeah, research in the deep sea is
complicated.
I did some freshwater ecology before, and it's not the same.
All right. I don't think we have any other questions that I can see. So I will just thank you both for your talks. I'm hoping I'm showing the right screen. Are you seeing the screen with your faces on it? Good. Just to say that our next webinar is next month, Thursday the 17th, and it will be by Luisa Barrera and David Liberti on microalgae working for a cleaner planet, a completely different science, valorizing waste, resource recovery, and circular innovation. It is based, linked to the work that we did in our document on Navigate the Future 6, and is part of the remediation project. So with that, I just want to thank you both for a very fascinating discussion. It was really interesting to see the science you do. And very important science in both cases. I think both of these things have very good application. So well done. Thank you.
Thank you.
Bye.